Hello everyone, today's class will be on DNA and genetics, and my name is Imelda. Classes we remember that the cytoplasm is a base substance that contains all organelles and allows for metabolic reactions to take place. Ribosomes are small round organelles which are mainly found attached in the plastic reticulum or free floating in the cytoplasm, whilst the nucleus controls all of the cell's activities. Now, still on the nucleus, we found that within the nucleus, there are nucleic acids, which are large biomolecules essential for all life, and they store and transmit genetic information. There are two main types, DNA and RNA, and they are made up of building blocks called nucleotides. Now, the structure of the DNA was firstly discovered by Rosalind Franklin, who took pictures in the 1930s using X-rays, whilst James Watson and Francis Crick discovered the double helical structure in 1953 using Franklin's pictures. Now, like I said earlier, there are like I said earlier, both DNA and RNA have nucleotides, so structured towards the left of the screen. DNA and RNA both have four different nitrogenous compounds. They both have adenine, guanine, and cytosine. However, the difference between the two of them is that DNA has thymine, whilst RNA has uracil. Now, it's important to note that guanine always pairs with cytosine in both DNA and RNA. However, in DNA, adenine pairs with thymine, whilst in RNA, adenine pairs with uracil. Now, the structure of the DNA, the pairing of bases in the DNA means that two strands of DNA are joined together, forming a long ladder-like structure. The nitrogenous bases are held together by weak hydrogen bonds, and the ladder-like structure shown in the image on the screen becomes called and is known as a double helix structure. Now, the DNA strands wind around proteins, which are called histones. Now, if you look at the structure, we can find the sugar phosphate backbone, the weak hydrogen bond between the two complementary nitrogenous bases, as well as the different nitrogenous bases. Now, the DNA has three main functions. To control the function of cells, to regulate the functioning of genes, and to pass on hereditary characteristics. Genes are a unit of heredity, and they are responsible for the coding of proteins. They're also responsible for the phenotype of an organism. Yes. Now, looking at the RNA, the structure is shown on the screen. We have a single strand with several nucleotides containing the phosphate groups, nitrogenous bases, and the ribose group. Now, the location of the RNA is also dependent on its function. The messenger RNA is formed in the nucleus, where it enters the cytoplasm and attaches to the ribosomes. The ribosomal RNA is found in the ribosomes, where in the ribosomes in the cytoplasm of the cell, whilst the transfer RNA is found freely in the cytoplasm of the cell. Now, we'll come back to review these RNAs because they are important for the synthesis of proteins. Now, for a quick quiz, on the screen you have structures. I need you to identify all four of those structures, and then we have nitrogenous bases. I need you to tell me what those nitrogenous bases pair with. I'm going to give you a bit of time to do that. Let's go. So it's time for us to review our answers. So we have number one being the nitrogenous base, number two being the deoxyribose sugar for the DNA, the next one being the phosphate group. We have the structure of the circled part being a nucleotide. We have guanine binding with cytosine and adenine binding with thymine. Now we're going to look critically into the DNA replication process. Now it's important to note that the DNA replication process comes in five steps. Firstly, we have the unwinding and unzipping, where an enzyme known as helicase breaks weak hydrogen bonds between base pairs, separating the two strands of DNA molecule in the parent cell. Then we have the forming of a template, where each strand acts as a template for the formation of a new complementary strand of DNA, forming two sets of DNA. Then the nucleotides pair up, where three nucleotides in the nucleus pair up with bases on the exposed single DNA strands. An enzyme known as DNA polymerase matches the bases on the template with the free nucleotides through complementary nitrogenous base pairing. Pyrimidine bases known as thymine and cytosine pair with purine bases known as adenine and guanine. Now, then afterwards, there's a formation of the helix structure, the double-stranded helix structure that we know, where one strand is twisting to form that helical structure. Now, DNA replication is a very significant process because it doubles the genetic material so it can be shared between the resulting daughter cells during cell division and may result in the formation of identical daughter cells during mitosis as well. Now, DNA replication can also have errors when the incorrect nitrogenous base attaches to the original strand and the nitrogenous base is added or deleted. The sequence of the order of the bases changes on the new DNA molecule. This further results in changes known as mutation. Now here is a quick quiz on the diagram of DNA replication. Remember, I said it occurs in 5 different steps. I would like you to try to identify all numbers here, and then we could review it afterwards. Now, moving on to DNA profiling. A DNA profile is a pattern produced on X-ray film. It consists of lines which are of different lengths and thicknesses and in different positions, and they are different for each and every person because we are all genetically different, genetically unique. Now, DNA profiles are used to identify crime suspects in forensic investigations. They prove paternity and maternity. They're being used to determine the probability or causes of genetic defects, to establish the compatibility of tissue types for organ transplants, and also to identify relatives. Now here we have an example of a DNA profile, and we have a crime scene sample, and we have 3 possible suspects. I would like you to give that some seconds and tell me who you think is the suspect, the correct or the right suspect. You probably guessed it right because it is highlighted with a neon blue or neon color. Yes, suspect 2 is the right suspect because we find that the DNA strands and the DNA bars are exactly the same with suspect 2 as it is found in the crime scene sample. So yes, suspect 2 is the criminal that we are looking for. Now, protein synthesis is a process by which proteins are made in each cell of an organism to form enzymes, hormones, and new structure for the cells. They are made by linking various amino acids that are present in the cytoplasm of the cell. We found that there are 20 different amino acids and they combine in a large variety of combinations. As you can see, in the diagram there, we have an amino acid chain, and it consists of several different types of amino acids, and the form or structure of those amino acids are shown there with the amino group, the acid carboxyl group, as well as the R group, and the hydrogen. Now, the bond between amino acids is known as a peptide bond. There are three consecutive nitrogenous bases on a DNA strand is known as a base triplet. Now, if you look to the image on the left, you can see that they have outlined three different base triplets for us. The base triplets determine the amino acid that will be placed into the protein, as well as the sequence in which the amino acids will be joined. Now, these base triplets not only they are important for the synthesis or the structure, yes, the synthesis of a polypeptide, as you can see below. Now, the protein synthesis is divided into two main sub-processes. We have transcription and translation. In transcription, firstly, we have a section of the double DNA that unwinds, and then the weak hydrogen bonds between the nitrogenous bases of the DNA break, and the DNA unzips. Now, one strand of the DNA acts as a template, and the DNA template is used to form a complementary strand of a messenger mRNA, which is the mRNA, and it is done using free nucleotides in the nucleoplasm. The mRNA then codes for the protein which will be formed, and like we said earlier when we spoke about the codons, the three adjacent nitrogenous bases will be known as the codons, and they code for a particular amino acid. Then the mRNA moves out of the nucleus via a nuclear pore into the cytoplasm, where it attaches onto a ribosome. Then we move on to translation, which utilizes the transfer RNA, the tRNA, in the cytoplasm, and it has three adjacent nitrogenous bases known as the anticodon. It's important to note the difference between the codon and the anticodon. Now, the anticodon is for the transfer RNA, while the codon is for the mRNA. Now, the mRNA's codon will be complementary to the tRNA's anticodon. Now, each tRNA will carry a specific amino acid, and the codons on the mRNA, according to those codons, the tRNA will bring the required amino acid to the ribosome. Now, the amino acids are linked by a peptide bond to form the required protein. Now, it's important to note the differences between transcription and translation and where they overlap. Transcription involves the formation of RNA from DNA, creating RNA copies of genes for the cells using biochemistry. It occurs in the nucleus, and it uses mRNA, tRNA, RNA, and non-coding RNA. Translation, on the other hand, synthesizes proteins using an mRNA template, synthesizes proteins in the cytoplasm, and also, yes, like I said, synthesizes proteins. Now, they overlap where in biological processes, and they both involve initiation, elongation, as well as termination. Now, a summary of the lesson. What we've learned so far is that the nucleus contains both DNA and RNA. DNA is double-stranded, whilst RNA is single-stranded. Both RNA and DNA have nitrogenous bases, adenine, guanine, and cytosine, while DNA has thymine, and RNA has uracil. Now, a gene is a unit of heredity as it codes for protein. DNA replication occurs through five main steps known as the unwinding and unzipping, the formation of templates, the pairing of the nucleotides, the pairing of the bases, and then the formation of the double helical structure. Also, furthermore, or lastly, protein synthesis occurs through transcription and translation. That has been today's lesson, and I hope to see you next time. Thank you and bye.